I want to wrap this up by coming back to breathing and the brain, and just talk a bit about how we think both oscillatory and non-oscillatory brain activity is linked to respiration. And I want to use that more as a background to introduce an idea that I think could be of interest to hopefully many of you. And I'm going to briefly touch on a few things that have been occurring on and off during both the summer school and the symposium as well, because Tani and also Martin have introduced the this broad schematic of what we think is going on in the brain when we say breathing is coupled to the brain, at least for the oscillatory part. So this is at least one of the mechanisms that we think is causing the effects that we see with respiration. So the very respiratory rhythm being generated in the brainstem, then propagating through thalamus and the cortex. At the same time, the signal goes down to the respiratory muscles and so on. But at the same time, we have, with every breath, we have the feedback with the airstream triggering neurons in the nasal cavity connected to the piriform cortex and so on. So what it comes down to is, at least for the oscillatory coupling of respiration and brain activity, is phase amplitude coupling, which means that the phase of a slower signal, respiration, modulates the amplitude of neural oscillations, which are faster in frequency. And one application of this you can do with EEG. We did it with MEG. So whatever neural time series you have, be it from a source localized voxer, from an MEG channel or an EEG sensor, you can basically do a time frequency transform and go across the whole frequency spectrum, compute the amplitude envelope of whatever frequency you're interested in this case, just all of them, because the question was, can we map those modulation effects throughout the brain in a non-invasive way? And then compute some kind of coupling measure to arrive at a spatial map of where in the brain is which frequency modulated how strongly by respiration. And you can relate those to different networks that we know from resting state recordings. At the same time, not everything that we see in the brain is oscillatory. So the other side of the coin is that whenever we look at power spectra of brain activity, we get this complex intermingled signal of both oscillations that we can identify as peaks on this very broad slope, meaning that the higher frequencies carry less power than the low frequencies whenever you look at brain data typically. And that has to do at least in part with the interplay of excitatory and inhibitory brain activity, for which different receptor types come into play, with typically excitatory receptor types having shorter refractory times, which explains their dominance in the high frequency, whereas the inhibitory activity is on the lower end of the frequency spectrum. And I want to point out a relatively new toolbox that was introduced by our collaborator, Luke Wilson, from Sylvain Bailly's lab, who actually found a computationally feasible way of using the classic FOOF paradigm in a time-resolved manner, so that you could use it to dynamically investigate developments of this 1/f slope over time, and in our case, over respiration phase, because the question is, if 1/f slope is some kind of index of excitation inhibition balance, does that change over the respiration cycle? So again, you take your neural time series and you compute at different points in time as you wish, compute both the aperiodic slope and your power spectra, and then you could do something like compare 1/f slope and power within a certain area of the network that we previously identified, And you could also go around the respiration cycle. So this upper half would be inspiration and here would be expiration. And then on the group level, in both EEG and MEG data, show which respiration phase brings along which changes in 1/f slope and oscillatory power of a given frequency. You can look at this on the whole network level and then see whether those differences persist, and they do. So we have different modulation characteristics. If you look at the respiration cycle of oscillatory and non-oscillatory activity, you can also investigate those, as Martin pointed out, for the pupil data in different regimes where you have people breathe normally or voluntarily deeply. You can look at nasal versus oral breathing, which takes away some of those coupling instances that we saw in the beginning. And I want to use this as a background to introduce an idea that we published a short while ago, where we wanted to address. We've heard so much about elephants in rooms. The biggest one for me is that none of us has any idea about how all those different peripheral signals interact with one another. So we have sort of had symposia on pupil, we heard about respiration, we heard about cardiac effects, but the body as one dynamic system with the brain on top of it that has its own thing going on, how can we actually bring some kind of systematic in there? So this meeting is focused on brain-body interactions, but then we have this whole external world that interacts constantly with the body. And the idea here is, and this ties to Elio's talk about dimensionality reduction, is that basically you can construct as many variables as you want. So each physiological signal, each factor, genetic predisposition, all of this could go into what makes you at any point in time. So however many dimensions you want to come up with, over time you basically form a trajectory through all of those different variables. And whenever we have a participant in the lab and we do some kind of MEG recording maybe, or an EEG session, or do some training with them, what we actually see is at that point in time, we have one configuration of all those variables. We just heard in one of the last few talks how, by Anne's talk, how mood and time of the day influence what we actually see. And our claim is that this is not yet accounted for in the way that it probably should be. So both external and internal variables all play a role. And the important thing is that you can characterize these states either as recurring, so that would be a very cyclic signal like respiration or the heartbeat. Some of these states will never occur again, so you'll never be as young as yesterday, right? So once we conceptualize whatever we observe in the lab as these brain-body states, we can then make predictions about how probable are upcoming states. So from where I am now, what are the probabilities for an upcoming state A, B, or C? What might my trajectory be like in a year or 10? And by incorporating those variables, we might be able to then identify trajectories that might be functional or also experienced as dysfunctional. And the idea is to record and incorporate in the models that we develop as many factors as possible. And then, as I said, coming to this dimensionality reduction idea, to then find a way to extract those factors that actually tell us something about the question that we have. So if we want to predict disease progression, or maybe inform choice of therapy, for example, all of these variables that we incorporate in our models will always be too complicated and too complex But what we could do is to try and quantify their interplay in a way that we reduce the complexity of the data down to a set of variables that is actually observable, and that we can use to inform interventions that could change the trajectory out of dysfunctional substates into functional future states. And with that, I'd like to close the symposium. Thank you very much.
One question, last question. We have a winner.
Okay. Thanks a lot. I love your vision. I think that's really probably the right way to go about it and look at all these things and see what's going to matter for what. Just out of curiosity, because what I showed before, right, with that the ECG aperiodic slope is also changing with some factors. Have you just by any occurrence looked into how like changes to your breathing rhythm would or could modulate that?
Sorry, I couldn't get the last half sentence.
Okay, sorry. Have you looked into by any chance how like differences in breathing rhythm could impact maybe some aperiodic features of the ECG signal?
I've never investigated 1/f in ECG.
Okay.
No. So just to get this right, this will probably be a multi-decade.
No,
right?
it was not about that. It was more about the previous study.
No, but what you're asking is exactly the right question, because we have to begin by identifying 2 or 3 factors that we can make sense of, and then move on from there, right? So whatever we do contributes to that bigger understanding and contributes to us developing a systematic approach to understanding all those complexities. This particular thing, sorry.